Hello friends, welcome to BMH Learning. In this video, we are going to discuss about phenol estimation by FC method. So in this tutorial, we will be talking about how the phenols can be quantified using Folin Ciocalteu method. Polyphenols have become an intense focus of research interest because of their perceived health beneficial effects. They have been reported to exhibit many effects like anti-carcinogenic, anti-atherogenic, anti-ulcer, anti-thrombotic, anti-inflammatory, vasodilatory, and last but not least, analgesic effects. Interest in the research of polyphenols has grown because polyphenols can be utilized as antioxidants in the food industry, and thus they benefit human health in various ways. The beneficial effects are due to their free radical scavenger properties that block the deleterious action of free radical molecules on cells. Assay principle. The FC assay has been used as a measure of total phenolics in natural products. The basic mechanism is an oxidation-reduction reaction. The FC reagent contains phosphomolybdic phosphotungstic acid complexes. The method relies on the transfer of electrons in alkaline medium from phenolic compounds to form a blue chromophore. The blue chromophore is constituted by a phosphotungstic and phosphomolybdenum complex. The reduced Folin Ciocalteu reagent is detectable in the range of 690 to 710 nanometer. The maximum absorption depends on the concentration of phenolic compounds present in the sample. Requirements: standard gallic acid, stock concentration 1 mg per ml. You can make 10 ml or 100 ml by dissolving gallic acid in distilled water according to the experimental need. Second thing we need is 20% sodium carbonate. It's because the reaction is carried out in basic medium. Dissolve sodium carbonate in distilled water by shaking the container. In case if it's not dissolved, give it a little heat. Next we need is FC reagent. Remember one thing here, the FC reagent is always stored in dark. Then we require plant extract. Any good solvent can be used for plant extraction. Let's say we are using methanolic plant extract. Procedure: add 100 microliter of plant extract into an Eppendorf tube. Into this tube, add 400 microliter of distilled water to dilute the sample. Give a quick spin to this tube to dissolve the sample in distilled water. Next, we add 150 microliter of FC reagent, diluted 1:1. Again, give it a quick spin so that the whole phenolic content should react with the FC reagent. Leave it for 5 to 10 minutes at room temperature. Bluish color compound is formed. Add 500 microliter 20% sodium carbonate. Incubate it for 1 hour in dark. Take the absorbance. Now let's do for the standard. So we are going to take different concentrations of standard gallic acid, that is 50, 100, 150, 200, 250, 300 up to 500, and the stock concentration we are using is 1 mg per ml. Now add distilled water in all different concentrations to make final volume 500 microliter. For example, in 50 microliter standard, add 450 microliter of distilled water. Give all the tubes a quick spin to thoroughly dissolve the contents. In all tubes, add 150 microliter of FC reagent. Quick spin all the tubes. Observe the greenish blue color. There should be gradual increase in the color intensity because of the increase in phenolic content in the standard. Now add 500 microliter of sodium carbonate in all tubes. Incubate it for 1 hour in dark. Take the absorbance. For blank. In case of blank, all the chemicals are used in similar volumes, but no standard or sample is added. This was all. Thanks for watching.